welcome to the last session of the 2025 edition of the RePo-Webinars, an initiative from the RePo-SUDOE project for the promotion of scientific knowledge and dissemination of technology for the drug repurposing in the SUDOE space. For those that joined for the first time these webinar sessions, the structure of the webinar starts with this brief introduction and also the introduction of the speaker of today. Then it follows the scientific presentation and some room for discussion and the final closing of the webinar within approximately 1 hour in total. So today we are very happy that we have Dr. Sergio Baranzini, a distinguished professor of neurology at the University of California, San Francisco. And Sergio will tell us about using causal, inference, and knowledge graph to inform drug repurposing. Before we start, just let me tell you some biographical notes from Professor Sergio Baranzini. So he earned his PhD in human molecular genetics in 1997 from the University of Buenos Aires, Argentina. His lab at University of California, San Francisco, uses a multidisciplinary approach to science, and it is composed by experimental and computational researchers. Dr. Baranzini is an active member of the International Multiple Sclerosis Genetics Consortium. He leads the iMSMS, an international consortium to study the effect of bacterial populations, so microbiota, on multiple sclerosis, susceptibility, and progression. In addition, he is the principal investigator of the SPOKE, a large multidisciplinary bioinformatics approach to gather, integrate, and analyze all biomedical data, which is currently supported by NIH and NSF in the United States. Dr. Baranzini received the Baranczyk Prize for Innovation in Multiple Sclerosis Research in 2024. So, as I told you before, today, Dr. Sergio Baranzini will speak about using causal inference and knowledge graph to inform drug repurposing. And without further ado, we are very glad to have Dr. Sergio with us today, and the screen is yours.
Thank you very much, Hugo, Antonio, and everyone at the RePo-SUDOE initiative. It's a great pleasure to be here and to be sharing some of our results with this community. So let me start sharing, and hopefully will work seamlessly. Do you see my full screen? Okay.
Yes.
Perfect. So what I prepare for today is, as you mentioned, Antonio, Hugo, my research is both in the computational field as well as experimental, and I'm part of several multidisciplinary collaborative groups. So what I thought I will put together today for this group is a little bit of a mix between the computational side and to share what are the things that we are thinking about and implementing. And then the second part of my talk will be an actual example of how we can utilize these methods to drug repurposing example in multiple sclerosis. So hopefully this will be of interest to everyone in the group. So let me start by describing a little bit of a philosophical, if you wish, concept, and that has to do with our immense amount of data that we have in the digital domain. Now, since the digitization of data and information in the early 90s, we as a society have accumulated a wealth of digital assets that now are for us to analyze in whatever capacity we might be interested in. And in this concept, I want to start by describing how we go from data to information and from information to knowledge. And then I want to make the distinction between those concepts because sometimes they're used interchangeably, but in my mind, I think that they're quite distinct, and that's important. So this sits on the work of Russell Acuff back in the late 80s, where he described data as facts and primary results of an experiment. So this is raw results devoid of any contextualization or synthesis. That's the primary results of an experiment. But then data can become information if we contextualize it, if we synthesize it, if we provide categorization of that. Not all data becomes information. Some data ends up in Excel spreadsheets and never integrated with anything else, but it can be synthesized and it can be contextualized, and then it becomes information with which, if we know what is the relationship between all those pieces of information, knowledge may emerge as a emergent property of that network of connected pieces of information. And I think this is very important because now, with the enormous amounts of data that we have in the digital domain and the power of our computational capacity, we can accelerate this transition from data to information and from information to knowledge. And let me bring you a couple of numbers that you may or may not be aware of on how we measure the data that we have in the digital domain. If we start at the top, we are familiar with the kilobyte. This is 1,000 bytes, 1,000 characters. Then if we go one order of magnitude above that, we have the megabytes. Another order of magnitude, we have the gigabytes. And then we have the terabyte, which is where we are now mostly dealing with in our daily lives. The petabyte, that's more enterprise type magnitude. Then we have the exabytes, which is another order of magnitude. And then zettabyte, and probably the yottabyte, most of you haven't heard, but it's what is next. And right now, if by some estimates, we've accumulated as a society roughly 64 zettabytes. And this was an estimate done in 2020. And the projection was that this will grow up to 175 zettabytes by 2025. So this is an enormous amount of information that includes everything that is our digital fingerprint of humanity. So this includes every single image, every single video, every single message that we send with our phones and so on, and every digitized book and so on. A couple of really sobering statistics is that more data was generated in 2017 than in the history of humanity prior to that. So 2017 was a hinge year in which, in that single year, we accumulated more digital information or data actually, than in the history of humanity. And then from there on, that figure starts growing almost exponentially. This means that 90% of the world's data was created in the last 2 years. And that was true in 2017. Now it's probably even shorter. So I like to think a lot about who has the biggest problem to solve with big data. So big data is everywhere. And of course, there's many aspects of life that are capitalizing on this enormous amount of data. And by some accounts, astronomy is one of the fields that is benefiting from this, where there's billions of galaxies in the visible universe, and we try to understand how these galaxies expand and how the exoplanets are being discovered, and this is an enormous problem that astronomy is benefiting from having this amount of data. Some may claim that the world of finance is benefiting a lot about this, and truly, with all the transactions that happen in the digital domain within banks, within clients, this is certainly an area of a very active research. E-commerce, of course, there's been an explosion of e-commerce in the last 2 decades that is impossible to ignore. But I will claim that biomedicine is the field that will benefit the most from capitalization of big data and interpretation of big data. Why do you think this? Because biomedicine is uniquely complex compared to all the others. The others are complicated. There's difference between complex and complicated. Complicated, the tax code is complicated, but biology and biomedicine are complex subjects that display many properties of complex systems, such as interdependence, such as emergent properties and dependence between variables, showing different properties that cannot be predicted based on the initial conditions always. And let me illustrate this point, that in our world, if we think about the hierarchical organization of biological complexity, we could start at the bottom, where information is basically generated at the genomic, or now you can claim metagenomic layer. This genomic information, then the next step up in the hierarchy is that of, well, transcription. So these sequences are converted into RNA. RNA is then translated into proteins, and then proteins interact in many different ways, giving rise to signaling pathways. So information is traveling through, not randomly, but through very well-defined pathways, which are still very complex. And then we have the cellular environment in which cells integrate all this information, and then they have a response. Cells integrate multiple cues from their environment, and then they have to respond to that complex set of signals with either a response such as proliferation or cell differentiation or even apoptosis. But cells in that regard are excellent integrators of information. And then if you continue going up the hierarchy, we have tissues, systems, and all the way up, we have the organism, we have the individual, that in our case is the patient that we will see in clinic. And unfortunately, all of these stages or hierarchies have been rather isolated in our pursuit of understanding the parts. So science has been extremely reductionistic in our approach, with good reasons, because we deemed that it was too complex of a system to understand in its totality, but therefore we broke it in smaller parts that we could understand. But what that caused is this siloing of knowledge, that rarely goes across this strata. And this is an extremely complex scenario in which the information spans 4 orders of magnitude in space and 5 orders of magnitude in time, if you think about from gene regulation to aging. And it's again, understandably so, extremely difficult to integrate and to put together into a comprehensive framework. And just to illustrate this complexity, this is just a metabolic map. So this is just one of those layers where information needs to flow. So many people have been thinking about ways of integrating, and our approach more than a decade ago was, well, how about if we start building a database of databases, what is known as a knowledge graph, that allows us to connect the different sources of information with semantically meaningful links or relationships, so that not only computers can understand, but humans can also interpret. So why a knowledge network or a knowledge graph? Well, first, to make use of the existing data, which, as I mentioned, is extremely abundant. And again, to balance this two scientific philosophies, if you wish, one of trying to reduce the dimensionality or the complexity to something that we can understand versus a holistic perspective and try to understand the whole rather than the parts individually. Again, I mentioned biology and biomedicine is uniquely complex. And to me, this is part of why medicine is behind the curve in capitalizing all these new developments that happen in the digital world, such as artificial intelligence and approaches in machine learning that usually take flights in other fields before they get to medicine. And in part, the success or the modest success that we've had in biomedicine is due to this complexity that we so far have failed to completely understand. So what is a knowledge graph? A knowledge graph, as I mentioned, is a database of databases. In its simplest form, is a graph database. And this is a distinction between most databases that you may be familiar with, which are relational databases. In a relational database, you have multiple tables that list different properties of the data, and then you link the tables via a common identifier. In a graph database, you have the ability to link multiple identifiers to multiple other concepts. So then this is a fundamentally different type of database in which searches are more powerful because you can identify related concepts, and you're making use of graph theory approaches to interpret the data. And in this case, I'm going to describe the Spoke knowledge graph, which is composed of, at this point, more than 75 databases, specialized databases that include genetics, proteomics, metabolomics, physiology, pharmacology, signaling pathways, even virus and bacteria signaling pathways that are of importance to humans, all the way up to food databases and so on. So the idea of this is that we can connect concepts. When here you see the word gene, so this is what is called a metagraph. So here I'm just showing the classes of objects, of the classes of concepts that we have in Spoke. So when it says gene, that means every single gene in the human genome is represented. And when there's a connection between gene and disease, well, that also means that every disease known to man is included, and their connections, if there is a known genetic relationship between a particular gene and a particular disease, we describe it with an edge, with a link of the type disease associates gene. So this is the semantic interpretation of this relationship. And we have hundreds of thousands of these gene to disease relationships. And then, as you can see, if you extend this to the whole graph, we have disease to symptoms. So any disease to any symptom recorded. We have disease to organisms. So any viral or bacterial disease. And then for each organism, we have their proteins that express. So we have more than 300,000 bacteria and viruses and their proteins. And then we know how their proteins interact. And we know how their proteins, if they are enzymes, how they are participating in metabolic reactions, and so on. So the idea of a knowledge graph is to try to connect semantically all the concepts that we as humans incorporate into our knowledge as we train in a particular field, but now it's computer readable, and we can take advantage of computer based approaches to analyze at a speed that humans cannot simply do. So this is a large collaboration that started at UCSF, like I mentioned, more than a decade ago. Now it involves several groups in the US and abroad, and is very active in many different fields. So today, Spoke is at the center of a number of initiatives that range from approaches to precision medicine, for example, which I'm not going to speak about today. Now we're connecting Spoke to large language models. We're collaborating with NASA. We're collaborating with different diseases, groups interested in different diseases. But our focus today will be how we're using Spoke to inform drug repurposing. And in particular, I'm going to center around multiple sclerosis because that's the field that I'm most familiar with. So, but let me tell you a little bit about how we're computationally taking advantage of a network such as Spoke. And this started several years ago in the lab when we adapted a method that was originally described for social science analysis. This is called link prediction in a graph. And this is a approach known as meta-path link prediction. And let me show you an example here in which we're using genetics to identify or to predict what might be a connection that we're not observing. And one of the advantages of metapath-based link prediction is that naturally embrace network heterogeneity. And as you can see, Spoke is a heterogeneous network because it has multiple types of nodes. In contrast to other networks, for example, the protein interactome, or a purely in other fields, you can think about the airport network or phone calls network or the actor network, which are homogeneous because all the nodes are of the same type. So this approach embraces naturally network heterogeneity. So this is one of the reasons we chose it. Another reason is that it's highly scalable. So it works well with hundreds of nodes or with millions of nodes. Then it really allows the implementation of supervised learning. now? Hello? Can you hear me now?
Yes, no.
Okay.
We lose you for a couple of seconds. Now we can hear you again.
Okay. Yeah, for a minute, I think it was muted. So, and then the final property of this approach is that it allows for interpretability. So it is not only, it does not lead to predictions that are a black box, such as many machine learning approaches. So it's interpretable. So how we approach this, and again, in this example, I'm going to show you how we use this to predict a link between a gene and a disease, in this case, multiple sclerosis. So one starts at the metagraph level up here in the top left, where there's, for example, genes connected to diseases based on known genetic associations. Genes are also connected to tissues based on their expression in different cell types. Tissues are also connected to diseases based on their co-occurrence terms. So if for example, in the literature for multiple sclerosis, usually blood or lymph nodes or T cells will be highly associated because that reflects the pathogenesis of it. And then genes are also connected to multiple categorizations, such as gene ontology classes and so on. So what we first do is we characterize the all the possible paths between a gene and a disease. So in this case, we know that multiple sclerosis is associated with a gene known as STAT3, also associated with gene CXCR4, IRF8, interleukin-2 receptor alpha, and so on. But the question that we're posing is RRF1 also associated with MS, given that IRF1 interacts physically at the protein level with these genes that are known to be associated with MS, and given that IRF1 is known to be associated with Crohn's, which is known to be associated with the STAT3, which is known to be associated with MS, and third, that IRF1 is known to be expressed in tissues that are involved in MS pathogenesis. Sorry, I think something happened here. Okay, we're back. So, so this is the framework. These are all the connections that we're trying to identify. Can we predict what is the probability that this gene is associated with MS, given that it is closely related in many different aspects to the disease? And for that, what we do is we compute what we call metapaths. So what are all the metapaths that lead to potentially IRF1 and MS? Well, one is through cell types. So IRF1, there's two paths that go from here to tissues, and then there's one path that go from the tissue to MS. Then there's several other metapaths that go through genetic associations and protein interactions. So here we have one metapath that goes from IRF1 to interleukin-2A to MS, and then so on, based on our data. So the approach takes advantage of all these metapaths. It downplays those that are extremely connected. So this is why it's called degree-weighted metapath count, because if some node is extremely well connected, it will downgrade that in the calculation, and then it will compute a final score that will be proportional to the probability that this gene is connected to MS. So with this approach, we were able to show that we could predict many genetic associations even before they were discovered. And that was very exciting, because that means that a priori, not every gene is equally likely to be associated with any disease. There is a hierarchy a priori based on the knowledge that we already have. And that's an important distinction between with the main assumption of genome-wide association studies, in which the assumption is that a priori every gene is equally likely to be associated. So this kind of departs from that, and it gives further power to identification of genes. So what we thought is, well, how about we implement this same approach towards drug discovery and repurposing? So our approach then took this form. Imagine that we have a number of small molecules, and here, if you zoom in, so this is a very zoomed out version of Spoke. If you zoom in here, you will see more than 2,000 small molecules that are known to be used in a number of diseases. But we delete those connections manually, and then we leave all the other connections with small molecules to their side effects, small molecules to the genes, the proteins, and we try to predict back these edges that we deleted. So it's the same problem, essentially. We're trying to do link prediction after we deleted the real indications, but we're going back from connections that we know happen, and then we ask, how well can we recall the known indications? And so, in summary, of this pipeline is an adaptation of the pipeline that I showed you before, but we first use a version of a Spoke. In this case, the reference that I'm showing here is several years ago, in which Spoke was rather much smaller than it is today. But the idea remains the same. We extract features from the network to quantify the prevalence of a specific path between each compound and each disease. And then we transform that into a probability by feeding a regularized regression model. And then we permute the network. So we shuffle the connections, and we repeat this several times to get sort of a default or a randomized version of the network to make to know what the difference is between the real network and the randomized network. And so with doing this, we got an extremely high prediction performance in terms of re-identifying the known indications. So if we use the most strict of the databases to predict which of these drugs that we identify in the top quartile of the predictions are disease modifying, we almost recover 98% of all the drugs. This means that there were some drugs that had high probability of link with the disease, but they weren't a known treatment. So those are excellent candidates to test in a drug repurposing approach. So let me now fast forward to multiple sclerosis and how we implemented this approach to identify potential candidates that will work in multiple sclerosis. So a very brief description. MS is a chronic autoimmune disease of the central nervous system, in which the myelin that ensheaths axons is targeted by immune cells and then destroyed. What this produces is neuroinflammation and ultimately axonal death and transection, which causes a number of symptoms, including visual loss, muscle weakness, numbness, imbalance, fatigue, and so on. And it typically strikes between the early 30s to the early 40s. There are multiple subtypes, and the most common subtype is a relapsing, remitting sort of disease. It affects almost 3 million people worldwide. It has a 3-to-1 female to male ratio, like many other autoimmune diseases. And there are known genetic and environmental factors. One of the main issues with MS is that there are existing therapies that are very effective to control the relapse and remitting course, but no therapies currently exist to address the progressive phase of the disease, in which once axons have been demyelinated, no drug has been shown to be effective to remyelinate those axons. So this was our goal. So a couple of more slides on MS. As I mentioned before, it's a combination of genes and environments, like many other complex diseases. But at the pathogenesis level, what we think happens is a antigen-presenting cell will activate a immune cell, a T cell, through presentation via the MHC of some unknown antigen as of today to the T cell receptor of certain T cells, and this will cause these T cells to become activated and then circulate. And like many T cells, when they circulate in the activated form, they will transvasate the blood vessels into every single tissue. They will produce a number of proteases, and including in the central nervous system. So when they gain access to the central nervous system, these activated T cells might re-encounter the same antigen or a similar shape of that antigen that was originally activating it in the periphery, but now in situ in the central nervous system. What this will cause is T cell reactivation with the production of a number of cytokines and chemokines that will further attract more inflammatory cells, that will activate local brain microglia and macrophages and astrocytes, that some of them will produce toxic chemicals, including glutamates, but also oxygen radicals, and altogether will cause this damage to the axon and will expose the axon to further damage. Then we know that there's also the contribution of B cells with antibodies, that altogether T cell plus B cells will cause this axonal damage. So, sorry, I pressed the wrong button. So ultimately, this axonal damage will lead to pathology. And if we look at the pathological staining of tissue, what we see here is a brain slice stained with hematoxylin and eosine, and you can see a transection of a blood vessel with plenty of macrophages and T cells emanating from it into the brain parenchyma. And this will translate into an MRI, which is what we actually see in patients, and this is a demyelinating lesion. So how can we utilize all the approaches that I mentioned before towards trying to repurpose a drug that will target remyelination in individuals with multiple sclerosis? So this is a project that was called Brave in MS, and this was a 4 million euro Progressive MS Alliance project, a network of several European centers and one Canadian and one center in the US to employ a repurposing approach to identifying drugs that could be used to remyelinate. And my colleagues, Gianvito Martino from San Raffaele in Milan, Marco Salvetti and Maria Piebracchio in Rome, Jack Antel in Montreal, Anik van Everkoren and Rahim Nahid Ousmar from Salpêtrière in Paris, Vincenzo Suma from a company called IRBM, Tanja Kuhlmann from Münster in Germany, and Frauke Zipp from Mainz in Germany. So this was a network, again, designed to first use Spoke at the very beginning to prioritize which drugs might have higher probability of having remyelination potential. So this was the first in silico part. And then the idea was to then, in the first year, to test those compounds for toxicity. Later on, see which one of those will penetrate the blood-brain barrier, because that's a required condition. And then which of those will have a remyelinating potential, and ultimately test that in animal models to end with a clear selection of very few compounds that then can be tested in humans. So in this approach, as a summary, and I'm going to show you some details, but in summary, we started repurposing, categorizing about 500 drugs, which we identified with Spoke, and reduced that to a more manageable number that could be tested in first vitro and then in vivo. So basically, this is the selection process that happens at the beginning, using a very similar approach to the one that I described before. We take every possible compound that is known now to be approved in either the US or Europe, and then based on the paths that we know, which one is the most likely of compounds that will have an effect on molecules, proteins, or cellular processes that have to do with the myelination process. And again, we utilize the metapath identification system that I showed you before, computed degree-weighted path counts, and resulted in a smaller set of molecules that then we took on the experimental side to verify which ones might have the greatest potential. And the first thing that we did with this was to evaluate what was the octanol water, so the solid, the organic to aqueous phase partition coefficient. We incorporated measures of which might be the ones that have the best blood-brain barrier penetration, which cells might be affected by that, and we, of course, wanted to predict central nervous system activity. So this is some hard data showing that these are the 273 compounds that were prioritized and that were tested. And first, we tested their toxicity at different concentrations in different cell lines, including brain cell line from rats, but also primary oligodendrocyte precursor cells. Oligodendrocytes is the glial cell that produces the myelin. So this is, we want to preserve these cells, so we want to make sure it's not toxic. We also tested this in neurons to see that it's not toxic to neurons. And we utilize technology that will use first primary neurons, but also the pluripotent stem cells and neurons that were derived from fibroblasts from patients. So by using all these approaches, we reduce the potential drugs to only 32 top runners based on their toxicity profile, and we carried on with these 32 drugs. The next idea was to select drugs for their neuroprotection potential. So we want drugs that are both that can remyelinate, so that can induce differentiation of oligodendrocytes, but also that can act on neurons and increase their resistance to damage. And the assay was as follows. We culture neurons that were iPS cell derived, and we then put them in culture which each of these 32 compounds at a 1 micromolar concentration. Then we challenge the neurons with NMDA to produce a highly challenging toxic environment and see which ones survived. And then based on this, these are our positive controls. Based on this, we can identify which ones are favoring neuronal survivability on these challenging conditions. Then a different assay was, again, as I mentioned before, to identify the promyelinating effect of the same 32 drugs. We tried different systems in which these drugs might help produce that maturation of oligodendrocyte precursor cells into mature oligodendrocytes that can myelinate. So using 3 different systems, one from rat oligodendrocytes, one from humans, and another cell line, then we identify which are the compounds that have activity in all 3 systems, and then we started to really narrow down the field. We also produced information on the promyelinating effect of the lead compounds, and you can see that here there's 2 compounds, bavicent and casopitant, that produce the most myelin. So the green staining here is MBP, myelin basic protein. This is the basal condition, and you can see how bavicent and casopitant are increasing the signal for MBP considerably in 2 different systems. This is myelinating neurons, and this is myelinating nanofibers. This is a system in which nanofibers of plastic are being cultured with oligodendrocytes, so the oligodendrocytes will wrap around. And we see that there's an increased effect of particularly one of these drugs, bavicent, in these 2 systems. And the final step was to test this in preclinical models of MS. The typical preclinical model of MS is called EIE, for experimental allergic encephalomyelitis, in which myelin or myelin components are injected into mice and then inducing an autoimmune response. So 2 weeks later, the mice develop demyelination, which can be then pathologically analyzed, like in this case with electron microscopy. You can see how the axons that you see here in a transverse view get remyelinated. So myelin is this concentric thick border that you can measure that in this compound reduces the G ratio significantly, and the G ratio is the thickness of the myelin divided by the thickness of the axon. So you can see how much this compound remyelinates in vivo in this mouse model. And then finally, trying to understand the mechanism of action, what is bavicent doing to cells? We did RNA-seq to first of all, see that it has a clear signature compared to medium alone. Many genes are upregulated, many more are downregulated, but among the genes that are upregulated, several of them have to do with myelin production. So this is a gene called MBP, like I mentioned before, myelin basic protein, myelin-associated glycoprotein, CMP, MOG, and so on. So these are all genes involved in oligodendrocyte biology that get upregulated with when cultured with bavicent. So finally, what is bavicent? Bavicent is normally being used in clinical trials to treat alcoholism, ADHD, and attention deficit disorder, but has never been tested in the context of remyelination. So we think this is a very good candidate, and we're pursuing this now into clinical trials with in humans. So I'd like to stop here and summarize what I've shown you today is that knowledge network-based representation is a powerful paradigm for multimodal data integration, and we think that that's a very powerful approach. Spoke itself is the first-in-class biomedical knowledge network. Applying machine learning approaches on Spoke can help prioritize drugs for repurposing. We cannot test all the possible drugs with such complex approaches, like I showed you, going from toxicity all the way to effectiveness in preclinical models. So we really need to narrow down the possibilities, and here's where Spoke can help. And we ended up with the identification of this compound as a drug with potential for progressive forms of MS. So I'd like to thank all my collaborators, and both at UCSF and in the Brain MS Consortium, and our funding agencies, and I'll be happy to take any questions. Thank you so much.
Thank you very much, Sergio. Thank you very much for this very comprehensive view and presentation of how to get from the data to the information and to the knowledge within Spoke related to drug repurposing and to the multiple sclerosis. So thank you very much. So I will open the session for questions from the participants. Please go ahead. If we have questions from the participants, of course. If you want, you can raise your hand, open the micro, or write in the chat. I have a question, Sergio. So you spoke well, you spoke a lot about the Spoke database. So it includes 75 databases with very different kinds of data. So how can we use the Spoke research when we use it? How can we make knowledge from so different kinds of data? We are able to select which data to relate with a different kind of data, or this is internally managed by the research Spoke, and then how the results that we can obtain, how can we validate them?
Yeah. So Spoke is an open resource. So if you go to spoke.ucsf.edu, there's a web page where it will take you to more information about how can be used. There is a web interface where anybody can search for a particular concept, a disease, a gene, a protein, a molecule, and then download the immediate neighborhood. You can select what type of information you want to display. Of course, there's limitations as to which how complex you want your network to become with millions of concepts and hundreds of millions of relationships. It's impossible to visualize it all. But if you have a hypothesis, if you have a concept of interest, and you want to know what are the first neighbors, the immediate neighbors of that concept, or the second neighbors, and so on, you can do that. Then there's an API that people with programming knowledge can also query Spoke in an API fashion. So how to validate? Of course, that's the key question, and that we cannot replace experimentation. So experiments will still need to happen. But what Spoke can do is to provide that confidence that or that source of knowledge that it will increase the probabilities perhaps of your experiments being successful. You're not completely blind because you're considering many aspects that are hidden perhaps, but if you do this approach, you can increase your chances of success experimentally.
Okay, thank you very much. Not sure if we have further questions from the participants in the session. I think I make all my questions at once. In the regarding the candidates that you obtained using Spoke, you obtained 273 candidates that you still have to test experimentally. That's right. That's a huge work.
Yes. Again, it's we wanted to be comprehensive, and if we wanted to be comprehensive, we started from more than 3,000 drugs. So 3,000 drugs were initially narrowed down to 200. 200 is something that it can be tested. Of course, the goal is, can we go from the 3,000 to 2 and then just test those 2? I think that now is still the goal, but we wanted to make sure that we didn't leave any candidate out. So that's why we proceeded with first screenings only with the selected compounds, with the top runners, and then make sure that that we had a good representation.
Yeah. Okay. Further questions? If not, I will not do more questions. So just a final remark. Thank you very much for the participation in this RePo webinar cycle. Good luck for this Brave in MS initiative. It is very multiple sclerosis is a very strong disease. So actually, drug repurposing it's as the possibility of achieve a significant treatment for the disease. So I would like also to thank to you, Sergio, for this presentation today, and also for the thanking for all the speakers and the participants that were involved in this RePo webinar series during the year of 2025 so far. And so thank you very much.